Good afternoon. Welcome to FSHD University. We are your place for learning everything about FSHD, from living with the condition to the latest research. We're every month on the third Thursday, usually at 1:00 p.m., but today we made it later, 2:00, to accommodate Dr. Perlingueiro's schedule. Actually, we bumped it back a week as well. So sometimes it changes, but usually it's on the third Thursday of every month at 1:00. And you'll find it on our calendar and in our email newsletters. So with that, I would like to introduce our speaker today, Dr. Rita Perlingueiro from the Lillehei Heart Institute at the University of Minnesota. And today she'll be telling us about her laboratory's research on pluripotent stem cell research and FSHD. So over to you.
Thanks, John. Thanks, the FSHD Society, for the invitation. It's nice to be back. I was looking my talks, and last time I presented was June 2020. So it has been a while. A lot has happened since then. And I'm happy to share what we have been up to in the last almost 3 years. So let me see here. Yeah, so this is just an outline of what I will be discussing today. A little background on pluripotent stem cells, what they are and their capabilities. I will be splitting the talk in two topics. Our recent work on the therapeutic potential, or the regenerative potential of pluripotent stem cell-derived myogenic progenitors in FSHD, and our translational efforts to bring a pluripotent stem cell therapy to the clinic. I have to disclose that I'm a co-founder of Myogenica, is a University of Minnesota startup company. So pluripotent stem cells, let me just get, I will use my mouse to point to what I want to show something in particular. So pluripotent stem cells are this type of stem cells that have the capacity to generate all the cell types of the body. So different from hematopoietic stem cells or muscle stem cells that just have the capacity to generate a cell type within that tissue. So pluripotent stem cells have this amazing capacity to be expanded in the dish for a long time, and in the presence of the right conditions, you can tailor the differentiation of these cells toward the lineage of interest: cardiomyocytes, neurons, islets, hematopoietic or blood, or skeletal muscle. That's our tissue of interest. So pluripotent stem cells, in particular, we are interested in induced pluripotent stem cells, which are these cells that are reprogrammed from any cell type of the body and induced to a pluripotent state. So these are cells that I have introduced last time because of time. I removed those slides, but you expose any type of the body to this cocktail of growth factors and become pluripotent. So that's why we call induced pluripotent stem cells. So in this scenario, you can get cells from a patient with muscular dystrophy or from a healthy donor, generated iPS cells. If in case of the patient, you can perform gene correction and have gene edit cells that you can use for potential treatment. And you can also use these cells for study the disease in the dish and also for drug screening. That brings us to different aspects of research that you can take advantage of pluripotent stem cells. So we do all of this, but I will really focus on the main area of research in my lab, our main interest, which is in vivo regenerative potential, and based on what we have observed in the last 20 years, the development of a cellular therapeutic from these cells. See, it's not going. Slide control. Okay. All right. So just to give you an idea, it sounds almost science fiction, right? That you can reprogram your cells and it can become anything, and we actually can use the cells that can generate all the cell types in the body. These are actually being used in the clinic and they are being shown to be safe. So there are a number, and I just want to illustrate a couple of diseases that are being tested in the clinic and so far have been shown to be safe and have some efficacy. So diseases include macular degeneration, Parkinson's disease, in particular solid tumors and hematological diseases. People have derived natural killer cells to treat tumors. So these are more advanced in the clinic. I would say the solid tumors and macular degeneration, but Parkinson is also has a couple of clinical trials that just, I think they're starting in US and started in Japan and Europe. So these are diseases that it's important, you needed to generate the cell type of interest, the differentiated cell, in the case of neurons or NK cells, and you need to generate large numbers of these cells. And once you transplant, you generate your functional, the cells will produce their function. The case of skeletal muscle, there are a couple of things that make it more challenging. As you know, muscular dystrophies are very heterogeneous. I just have this cartoon showing that different muscles are affected. So depending on the disease, you want to target different muscles. But what makes it, what I want to illustrate in this slide, as you can see, this is an unaffected biopsy. Here you have the muscle fibers, everything is looking good. This is an example of the muscle degeneration of a patient with Duchenne muscular dystrophy. Of course, among different types, you might see this a little bit different, but in the end of the day, they all culminate with muscle degeneration. So just as an example here, in the early stage of DMD, you're going to start seeing a lot of inflammation. The muscle fibers are being destroyed, and you have starting to see fibrosis and a lot of inflammation. And by the end stage, a lot of fibrosis, and you don't really see the nice myofibers anymore. What I really show in this slide here today, and I always show this slide, is I want to point out that we have these myofibers that have this purple dot here, which is the nuclei in the middle. And what does this mean is that the muscle is regenerating in response to the degeneration due to the muscular dystrophy, right? So here they are all in the eccentrically located, and here they are mostly centrally nucleated. This means regeneration. And the regeneration is coming from muscle stem cells. So why am I saying this? What I'm trying to convey here is that when trying to develop a stem cell therapy or a cell therapy for muscular dystrophy, there are two cell types that you want, to produce. You want to produce muscle fibers, the myofibers that you can see here, and you also need to generate stem cells because the muscle has stem cells, you need to produce the cells also in vivo so they are able to provide long-term regeneration. Okay, so can pluripotent stem cells do that? So this is just a very brief summary of the work you have done in the last almost 20 years. It's my anniversary in 2023. Pluripotent stem cells we have used, as I said, they can generate any cell type. So our strategy is to use these transcription factors, PAX3 or PAX7, that are very important to tell a cell to make muscle or not, very early in the lineage production. To produce very large numbers of these cells. And this is a system that doxycycline inducible, so you can genetically induce the cells to produce large numbers of these stem cells, or we like to call progenitor cells, in the dish. And once you transplant them into animal models of muscular dystrophy, they produce myofibers, muscle engraftment, that's in red here. They also produce stem cells, is what I'm trying to indicate here. They are located under the basal lamina, the periphery of the myofiber. This is a donor-derived fiber, and they improve the function. So here in the left, I enumerated the main aspects we have seen, and I put an arrow on the important aspects that you have to keep in mind if you want such cultures to be used to develop a cell product. You need to really a robust expansion of the cells because you're going to need millions or billions of cells. And we want to make sure that these millions of myogenic progenitors, they are homogeneous, right? You don't want cells that are too differentiated or undifferentiated. So you have to reach really the sweet spot in terms of the cells, the target cell population. That's very important. Of course, coming with that aspect of the right population will have to fulfill three tasks. They have to make the myofibers, they have to make functional muscle. So these myofibers have to be there, but they also have to be healthy. Or another way of showing the idea is that they're going to make the muscle stronger. And another aspect that's important for the long-term potential of this transplant is that they also make stem cells upon transplantation. And we have done lots of studies in the last several years that show they can fulfill these three aspects. So before I move to the next step, this was just a little background. What we have done so far, well, we have tested the potential of mouse and human pluripotent stem cell-derived myogenic progenitors using this strategy in numerous animal models that we have published over the years, mostly in mouse models of Duchenne muscular dystrophy. We also had published on limb-girdle muscular dystrophy 2A, 2I, cells from unaffected iPS cells, also in the context of gene correction, and they all do the job. Then we stop and say how all these are recessive types of muscular dystrophies. So what happens in the case of a dominant type of muscular dystrophy? More specifically, as I talked to June many years ago in a conference which triggered this project that we completed last year, how about FSHD? What would happen if you transplant stem cells in the model of FSHD? This is something actually we wanted for a long time, but we are waiting for the animal model to perform these studies. And this work was led by a very talented postdoc in the lab, Karin Azag. I don't need to introduce the disease to this crowd. This is just for restate there. It's caused by expression, the FSHD is caused by expression of a normally repressed transcription factor, DUX4. So to perform this study to understand whether cell transplantation would have a therapeutic benefit, we used an animal model developed by my colleague here at the University of Minnesota, Michael Kaiba. He developed this mouse model that was published several years ago, in which DUX4 get expressed in this animal upon treatment with doxycycline. So we give this dox to the food, to the chaw, and then DUX4 gets expressed. These images are from his paper, as you can see. As you can give doxycycline, you see DUX4 expression. So this induces the expression of the DUX4. And as you treat these mice with doxycycline, you can see fibrosis, which is one of the main hallmarks of the disease. So we had the model to study. So how can you evaluate? So we have to come up with the best or different approaches to test our cell therapy. So the first set of studies, we didn't know exactly if only we would just induce DUX4 and that would be sufficient. So we usually injure the muscle. All these different animal models that we have studied regenerative potential on top of the disease phenotype of lack of dystrophin or lack of calpain-3, we induce injury because the injury makes the muscle even more damaged. So you can see even more the effect of engraftment. So since we didn't know exactly how this would pan out in this model, we had two arms. We had this mouse model. We treated or not with cardiotoxin, and we treated actually all the mice with DOX 1 week prior to transplantation. So basically in this week, we are inducing DUX4 expression. So when you transplant the cells, DUX4 is there. And you want to see if the cells 4 weeks later will have engrafted or contributed to muscle regeneration or not. And as you can see here, we also kept doxycycline for 6 days after the transplant. Okay, so the cells were exposed to doxycycline, or DUX4, I should say. So as you can see here, so we have a group that was just PBS, and another group that received, two groups that received cells with or without further injury. And as you can appreciate, this we are looking here in red, these are the donor cells. They were labeled with a red fluorescent protein. So just for simplicity, I'm calling them donor. Then we are looking where they are in the myofiber, and this is the nuclei. As you can see, they are all in the myofiber. And we have a large number of myofibers in both groups, with the presence of further injury. And this is something we had observed before with other animal models. So this is not a surprise. But it's important to notice they are there in both groups. So the cells do engraft in the presence of the DUX4 and in this animal model. So the next question we had, this is slide movements is giving me trouble. Okay. So before I move on, another aspect, you want to confirm that these cells, even though they are inside of the nuclei, you want to confirm the muscle identity. So we use a different marker, which is a myonuclei marker called PCM1. So we just have to appreciate that the green, that I have all those arrowheads there, they colocalize with the donor. So all these cells that I see that are from the transplant, they are muscle. That's the message here. So moving forward, we see the engraftment is all great. Then we start like, okay, the mice were exposed to DOX for about 2 weeks, right? How about if you reintroduce DOX? Because you know that in a muscle with FSHD, it's not going to be in and out, right? It's going to be constant. We couldn't do constant because the cells are also doxycycline inducible, and that's not good for the cells. But what we could do is to transplant, give some time, and reintroduce doxycycline to these mice and have DUX4 expression. And that's what you're seeing here. So we have a group that just have DUX4 induction prior to transplantation, and these two other groups that saw DUX4 again a week prior to harvesting the muscles and 2 weeks. So basically this group here was exposed to significant to DUX4. What you really want to know here, why are you doing this? The question here is whether that muscle that we saw in the previous slide that was donor-derived, if this DUX4 expression is going to perturb or damage this donor-derived healthy muscle. That's the question you want to address with these studies, because that would be the scenario that you would be observed if you were going to transplant this in a patient, right? DUX4 is not going to disappear all of a sudden. So it's going to have the healthy cells will be exposed to the toxic DUX4, and you want to know how they will behave in the presence of this toxic DUX4. So this is the example of the 7+7. So as you can see, we still see a large number of donor-derived engraftment in these muscles. This is a low magnification, so you can have a sense of, the big picture here. So dystrophin here is just to show you the myofibers, and this was really a large number. Well, so when you quantify this, you can appreciate that there is basically no difference when the muscles were re-exposed to DUX4, the engraftment is still there. So, and it's all higher than actually when the mice are exposed to DUX4 just for a week prior to transplantation. Almost, I think we have evidence from this publication that I'm not going to go in deep details here because it goes deep into the science, but is that this is stimulating the regeneration of the donor-derived cells, because you can see more engraftment when you re-expose the mice to DUX4. And this is from donor, right? So this is another way of looking at. So we counted the number of fibers, but here we are looking at the whole area, right? And it's the same idea. You're just another way of quantifying the engraftment. The other previous slide, we are quantifying the number of myofibers. Here we are using imaging tools to actually image the area that's positive for donor-derived engraftment. So we're pretty happy with that. And because we see that when we re-expose the mice to DUX4, we see more regeneration or more myofibers. This suggests that we actually have donor-derived stem cells, as we have observed in different animal models of muscular dystrophy. And this is the case, and this is what we are showing here. So when you are looking for muscle stem cell engraftment, we are looking for the presence of PAX7. That's what the green is showing here. And we confirming that this is co-localizing with our donor. So this is an example here of a cell that's positive for our red marker and is also positive for PAX7, a stem cell marker. We quantify this, and again, we see that that is, agreeing with the previous results, we have a higher number in those mice that were re-exposed to DUX4, which is a good thing. Okay, so this is really talking about the regeneration, but how can you know it's functioning against the disease pathology, which one of the main hallmarks is fibrosis. So we look at, and if you keep, let's just focus on the left panel here, which is the PBS, which is no treatment basically, right? So just inject saline in these muscles. So 7 + 14, just reminding you, these are the mice that were exposed 7 days before transplantation and 2 weeks just before harvest. And you can see it, this is what we are looking here in blue is the fibrosis. And as you anticipate more DUX4, more fibrosis, right? And you don't really see, you see very little in the mice that just receives for 7 days. And we are looking at 5 weeks later. But the mice that received DUX4, again, you can see there is significant fibrosis. But the mice that were transplanted, the mouse counterparts, that's the contralateral leg, you can appreciate here that we have less blue, right? So there is less fibrosis in the mice that received transplant. And this is quantified here, as you can see. So this suggests that the cell transplantation counteracts the DUX4-induced fibrosis, at least at this set of studies at these time points that we looked at. The next question, so I show you muscle engraftment, I show you satellite cell engraftment, and show less fibrosis. How about muscle function? So we are able to isolate, so what we did here to measure the force, we isolated the TA muscles that were transplanted, and you look at the in situ force measurements using this apparatus that you can see here is the Aurora. So we can see the isometric tetanic force here on the upper right. In red is the force measurement of a mouse of muscle that receives cells, and here is the PPS, the contralateral leg. You can see is weaker. But what is important really is calculate the specific force when you normalize the force measurement by the cross-sectional area. And as you can see here, the muscles that receive cells are stronger than PBS. So the conclusions from this part is that cell transplantation contributes to muscle regeneration in a model of FSHD, a dominant type of muscular dystrophy. So this is actually the first study to show that a cell transplantation can have a positive effect in the context of a dominant type of muscular dystrophy. Engrafted myofibers seem to counteract the DUX4-induced fibrosis, improve muscle strain. Of course, you have to keep in mind this is for this duration of the study. How that would behave long term is something that should be defined. Considering that the cells keep regenerating, the idea is that potentially will keep fighting with the fibrosis, and hopefully the healthy muscle will fight the sick muscle. Bursts of DUX4, and that's what I meant when you're doing +7, +14, right? When you are re-exposing the mice to DUX4, bursts of DUX4-induced muscle degeneration is stimulated the donor-derived muscle regeneration. And this work suggests that FSHD patients could also benefit from cell therapy if this becomes a cell therapy, which is the next topic of my talk. So switching gears. So after showing this proof of concept in recessive types of this muscular dystrophy and also in FSHD, how can you envision a potential pluripotent stem cell-based therapy for patients with muscular dystrophy? Well, there are two ways you can think of this. You can think of a autologous cell transplantation, and you have somatic cells from the patient with muscular dystrophy, regenerated iPS cells, you correct the mutation, you generate the myogenic progenitors, and you transplant back into the patient. In this case, we have one cell product going into one patient. Very exciting, but there are all the challenges associated with that. But it's possible, and we have data on that, and I'm just going to show you some references, not data today because I want to focus on the translational work. And of course, another way is to generate cells from a healthy donor, have these iPS cells to generate myogenic progenitors, and have an allogeneic transplantation, which these cells can go into multiple patients. So, well, for that, first you need to show that you can generate consistent number of myogenic progenitors, high number from human pluripotent stem cells or iPS cells. So this was achieved 10 years ago. We showed that using the same system, the conditional expression system of PAX3 or PAX7, you can generate myogenic progenitors that are highly expandable. Here we are comparing embryonic stem cells to two induced pluripotent iPS cell lines. That was the first proof of concept that actually they are similar. You can generate cells with similar ability from induced pluripotent stem cells, and they make myotubes. We transplant, again, this is first proof of concept, just to give you some starting point to where I'm going now. Here we are looking at these human cells into a mouse model of Duchenne muscular dystrophy. In this case, when you are transplanting human cells into an animal model, it's important that you have this mouse model in an immunodeficient background, because as you can imagine, mice don't like to receive human cells, right? So the level of rejection is very significant because it's different species. So for a better assessment of the ability of the cells, we like to cross the different animal models of muscular dystrophy to a mouse model called NSG that lack B, T, and NK cells. So then it gives us a free canvas in terms of the immune system. So we don't worry about the immune system, at least for performing these xenotransplantation studies. Obviously, when you're going to a patient, it's going to be the same species, but a different donor than you would deal with immunosuppression. But that's a different story. We can go there later. So for testing human cells into animal models of muscular dystrophy of disease, we like to use an immunodeficient background. And that's what we have here. And for looking at, very important, we need to rely on good antibodies. So we have good antibodies for human dystrophin. And as you can see here, there is no human dystrophin in a muscle injected, only saline. And we have nice areas of dystrophin positive myofibers in muscles that were transplanted with the human progenitors. And we don't like to rely only on one marker. So we also use human laminase C, which is also specific for human cells, not specific for human, but specific for, not specific for muscle, but specific for humans. So you combine human laminase C with human dystrophin, then you can confirm the identity of the cells that are muscle and two markers for human cells. So this is what we did here. We quantify and we see myofiber engraftment. We also look at force. The muscles of NSG, MDX mice, the dystrophic muscles are weaker. The muscles that receive only saline are also weak, and the muscles that receive cells are stronger. And we are also able to see that these transplanted human cells are able to make stem cells in these mice. Here we are looking again, PAX7 expression and the donor marker. Sorry, I didn't have time to actually change the labeling, the donor cells. Very important to look for safety, right? So we have a look at safety. These were mice that were transplanted for 11 months, which in a human would be several years. And we could still see engraftment, and we see no abnormality. And actually, we're following these mice for safety, but by almost 1 year, it's like, okay, let's see how the muscle looks like. Are the cells is still there, right? Which is always, important question. And indeed, the cells were there 1 year after transplant. And we have observed this again in several other studies, more recent. So again, back to that slide, my favorite. I told you we can do autologous. We have published 2 studies doing gene correction of limb-girdle muscular dystrophy 2I, and transplanting back in an animal model of these diseases is possible. We have transplanted in several studies in different animal models of disease, myogenic progenies from unaffected cells. So if you are going to move this to the clinic, what would be the best way? So this is something we, as a group, we discuss this a lot, right? Because obviously there is a lot of advantages of an autologous, but to start understanding the feasibility of a stem cell therapy, understanding feasibility for production, for quality control, so many aspects that I'm going to briefly go over with you, that we thought it would make sense to start with allogeneic transplantation. So an allogeneic transplantation, as I mentioned, is just you're going to generate cells from one patient that's from a donor that's unaffected, and we are able now to generate very large numbers of cells. And these cells, because pluripotent stem cells allow you to do that, and then you have in the freezer cells that can treat a number of patients. So if everything goes well, we actually all the high expenses associated with this production, I'll tell you, it's really expensive, allows you to maximize its application. While the autologous, exciting because it's patient-specific, one cell product is going to go to one patient, which hopefully is gonna happen in the near future. But we feel this is like second generation. There is a lot to learn with the allogeneic and then go to the autologous. Because once you know the production, all the manufacturing, all those aspects are worked out, adapting autologous. I wouldn't say easy, but much easier. So we eliminate all those variables also of adding gene correction, because it's another procedure that would have to be optimized clinical grade. So I hope I convince you that allogeneic is the best way, at least to start. To demonstrate the feasibility and safety of these cells, because then you can test a number of patients. So we started discussing with a group that I'll show you a picture in a minute, back in 2016. You know, time flies when you're having fun. So, I mean, I can't believe it, that it's 7 years ago. We started discussing what do we need to do to get this to clinical development. And in my mind, I was here because, you know, I told you had a lot of work done. I thought I was at least in the proof of concept, and my colleagues told me that I was actually in the starting point, the beginning of this era, really in the research and discovery, because we had a lot of steps to fulfill in terms not of so much the proof of concept, but actually in terms of the production that would be compatible with clinical applications. So lots of aspects. So this is the group that we got together back in 2016. So here we have experience with clinical grade production. That's David McKenna. He's the director of the Molecular and Cellular Therapeutics facility at the University of Minnesota. So we are very fortunate to have this facility right on campus. We have the Center for Translational Medicine that's directed by Robert Schumacher, which is also helping basic investigators like me to navigate regulatory and preclinical studies, IND submissions. And we have the expertise of cell transplantation. These colleagues here, they are really in the blood or the bone marrow transplants, but, you know, many aspects associated with enabling this to the therapy. They definitely had a lot of input and a lot of great feedback. So they're key components of this working group. And then we have the muscle expertise. Here, my colleague Michael Kaib and Alessandro Magli was a lab member that was really key in the first 4 years of this project. And then, of course, we have the neurologists. We can't move much without them. It's Peter Karashansky and Peter Kang that has joined us 2 years ago and will be leading our translational efforts. So this is a very long list that I had, and which is very summarized. But I'll need lists. I need to lists in order to see what's the task ahead of me, even though if it's, it's summarized. So I was really in the beginning. And one of the aspects we needed to make sure that we had clear, it was the methodology. Is this really the best methodology? Is this methodology safe? And the purification, because we want to make sure that the cells we are transplanting are safe and devoid of undifferentiated cells. So I'm not going to go into all these steps because most of them are published, or the first two are published. But I want to give you a sense of how much you need to do, right? So after you establish the methodology and the purification, you need to make sure that the production, all the culture conditions are compliant with clinical grade production means acceptable to being injected in a patient. So we have to eliminate or replace components that are not compatible with clinical trials. For example, horse serum, right? You don't want to inject chicken extract or things like that. You want to make sure that actually you can generate very large numbers, because I always say they expand a lot, but obviously in my lab, we don't need it to generate billions. So we always extrapolate. So when you have 10 million cells and you need to passage them, you just get a little bit of that and keep it percent, because I don't even have the space for that, right? Can you imagine how many flasks? So we optimize all this to conditions that were scalable, right? But you also want to make sure that once you scale up like that you still get the same functional cell product, right? Because sometimes you lose the function as the optimization process progresses. So you need, every step of the way, we are confirming that the cells were behaving the same way. Obviously, characterization of the product, and the list goes on and on. We had the pre-AND, so we had it very clear early on what the FDA would like to see. So the list is long, and we follow to the word. Then once you know you have optimized the method, you have to transfer the technology to the clinical grade facility, right? So you wouldn't want to do all this optimization there, because everything that's done there is really expensive. So we worked all the details of optimization in my lab, working closely with the team at the clinical grade facility to make sure that we were working towards the best methodology possible. And then they start working on the procedures, and then they start doing practice runs until they do the official run. We also need everything clinical grade. So we use lentiviral vectors. That was also generate clinical grade. And everything we do clinical grade is really expensive. So we generate the product clinical grade, and then we do the preclinical studies to have safety, biodistribution, toxicology. Then you do IND submission, and hopefully then you have the first clinical trial. Where are we in this list? We are right here. So basically, we have completed actually the GLP preclinical studies. We have followed mice done in a different facility for several months, and then the samples from these mice to confirm safety are done in a different lab. So at this point, nothing is really, I'm out of the game. I'm just coordinating and writing checks, but pretty much this is out of my hands and is done by tertiary labs. So these analyses are being performed right now, and we are expecting to get the data, the final results in May, and we'll be ready for IND submission in June, and hopefully with the first patient receiving the cells in 2023. And these cells are called Myopaxon. So here is just a snapshot of the production. So we have cGMP iPS cells. So this is coming from a unaffected donor that was actually generated by NIH. We produce APAX7 cells because that's how we make muscle, right? So this is third generation vectors, which are safer. We make a master cell bank, we call, and then we start the differentiation process. You purify with a published protocol, and then we generate a myopaxone. That's the myogenic cell product. This is just an example how the first initial study is just to confirm that the cells behave as you would expect. This is the morphology. They have a normal karyotype. So you don't want it. We check, this is just an example. We have a long list of safety precautions. And this is just one of the tests we do. These cells make myotubes in the dish, but this is just one of the assessment we do. The main assessment is in vivo regenerative potential. As you can see here, we are looking for the same markers we saw before. We are looking for human dystrophin and human nuclei or laminase C. So you're seeing green and red. So we see very nice muscle contribution after transplant. So as I mentioned to you, we have to make this all large scale, right? So when you're doing purification in the lab, you use FACTs or always small scale, right? So we use little tubes to purify the CD54 cells, which are the myogenic progenitor. So we have to move, because now if you're going to make this for patient application, we need to generate billions, right? So we are using this machine now called Clinimax, and they are specifically designed to generate cells for clinical trials, for clinical applications. So nice thing of this system is that all closed, as you can see here. So there is no exposure of the cells to the operator that's pressing the buttons here. Okay, so this is the the most favorable way of purifying cells by the FDA. Very important, the cells have the same expansion potential. I will show you in a bit. This is just to show that they, after purifying, they are still able to differentiate in the dish. As I told you, very important, how many vials do you need of this flask? Because you might need 10 incubators with this, right? So we optimize this to these different dishes, and you can see the growth of the cells is amazing. We can use the cells directly from the freezer, so they are off the shelf, which is a very important aspect for wide use, right? Because if every time you have to go back to the vial and make it fresh, you can anticipate the logistics, right? That I have a patient in Boston that wants to receive the cells, and we are making the cells, and the patient is scheduled to come in a Friday. Well, the cells need one more day. So there are all these logistics that make it complicated if you had to use fresh, although that's what we thought in the beginning. So we are pleasantly surprised that the cells directly from the freezer engraft as well the fresh. So the cGMP compatible progenitors are there at 2 months, they are there at 6 months, they are there at 1 year. So how all the process, I'm going to save you of all the details, but as I mentioned, there is all these SOPs, which is very detailed description of every single step of the procedures, right? So this is called a standard operational procedure. And let me tell you, it took months for this personnel from this clinical grade facility to get, because it's pretty complex process. So, but then it's important because then, he could do on his own. And it was amazing considering how many steps are there, that, it was done totally without our involvement other than the previous work, passing the details. It was all independent of our lab in the clinical grade facility. So that's very reassuring. So we had practice runs, and then we had official runs, which are actually the cGMP product that can be used in 2 patients. Just to give an idea, we got many vials of the master cell bank. We have the purified cells, and we have the final product in the freezer. We banked 150 vials at this number. So we have vials in the freezer to treat dozens of patients if the cells are proven to be safe. So we are following up on the stability, of course, right? This is an advantage of the cells, and people are not used to keeping cells in the freezer for a while because usually don't generate as many, right? So we are making sure that the cells are stable and functional over time. And these studies are ongoing right now. So this is just to show you regenerative potential practice runs, CGMP runs, pretty equivalent. So I was pretty impressed to see how comparable the productions are, which is really always scary when you're doing things in the lab, because this changes within people in the same lab, imagine in a different lab, right? So it was very reassuring. This is an efficacy when you transplanted the cells in that animal model of Duchenne muscular dystrophy. The engraftment is comparable to the other mouse models, about 200 fibers. And importantly, we see improvement in the specific force, as we had seen before. So where are we now? We are pretty in the last mile of this journey. So we are just finalizing to hear the results from biodistribution, toxicology, and hopefully we can wrap up the IND submission this late spring, early summer, probably late spring. So what are you planning for the trial? I'm sure you're full of questions about this. So the first goal here is really test the safety. You know, of course, we all want to see efficacy, but the main question is to make sure that the cells are safe. So to make sure that there is no side effect and no harm. And so if you detect that, then we can go in higher number of patients and in muscles that will bring benefit. So for that, we are focusing on the foot muscle of patients with Duchenne muscular dystrophy that are non-ambulatory. So if in the worst case scenario, we don't expect, but if something, if the cells have side effects, we are able to remove the muscle, and this will have no side effects to the patient. As soon as we confirm that the cells are safe, because of course we're going to have a biopsy, we're going to remove the whole muscle and you're going to be looking, because one is safety and another is to confirm the cells are there and doing what we expect them to do, right? So once we prove that the cells are safe and that is dystrophin in this case, we would like to propose to the FDA a second site for this group of patients that they can be benefit of a muscle injection in the hand muscle, which in these patients, as the disease progresses, the hand muscle is very important for their quality of life. As you know, this is like giving you the vision, right? I like to go step by step. It's hard to anticipate what's going to be the next steps without having the safety data. But what we envision is that once the safety is established, we will be ready to perform intramuscular injections in more muscles of patients with Duchenne muscular dystrophy and include other patients like limb-girdle muscular dystrophy and FSHD patients for muscle regeneration upon local injection. So additional muscle injection sites. Then, of course, one question that always comes most of the time is the systemic delivery. So we have to inject every muscle. So to start, it would be this way, because that every type of delivery requires its own package of safety. So we needed to begin safe, because you don't want to put this whole work and this whole potential therapy in jeopardy if you hurry up. So the idea is to go really, even though it's painful, to go in very small increments. I feel that we feel as a group that's the safest way, and hopefully we can get to many muscles target and many patients with muscular dystrophy, because the major advantage here is that this therapy, if proven safe and if efficacious, it can treat any type of muscular dystrophy, theoretically. At least, you know, one can say, are you sure about dominant? Well, we have proof concept for FSHD. So I think we can say that. So I'll end here because I want to have questions. I would like to thank the group. I have Alessandro here because he's no longer with us. He's in Sanofi, but he was instrumental at this work. Former lab members, they're contributing different ways for our knowledge and to the stage we are now. The collaborators, this is really amazing group of colleagues, Michael Kaiba with the FSHD project and Darko Bosnakovsky, the FSHD project, Michael Kaiba also in the translational project. And here are my collaborators that I showed before, the Indiana University with the production of the lentiviral vector for us, and also colleagues at UPenn with different analysis of our product. And of course, the funding agencies. Of course, I don't have all this funding at the moment, but I like to appreciate all the different foundations that have provide funding at different steps of this project. In particular, they led us to the point where we are to start testing these patients. But I would like to focus that the FSHD work was funded by the FSHD Society, and the translational work started with some funds from NIH and the Department of Defense, but was mostly funded by Duchenne UK for the preclinical work, and also funds from the Greg Marsoff Jr. and my director, very early funds from the parent association. And we had the discussions with NMED, and now we are having Myogenica to find investors for us. And I'll stop here, and I'm happy to take questions. Thank you for your time.
Thank you very much. That was an absolutely amazing tour de force presentation, not only of the animal model and the publication that just came up, and it was just really amazing to see in parallel all the progress that you've made towards applying these findings for the clinic. Thank you very much. That's great.
Thank you.
You've blown my mind. Because we knew this work was going on, but we kind of thought, the path to get to IND was going to be a bit farther away. Mm-hmm. And especially when you showed all the steps, and I really appreciate that you opened up the box and just what's involved, and that you got through all so far is absolutely amazing.
I still don't believe it that I'm in the last mile. Honestly.
Well, you're in a great institution, I think, to support that.
Definitely, it definitely is something you cannot do on your own.
Yeah.
Right. And you need people with the different expertise, and come together. We meet every month, and at some point it kind of develops, but deciding all those aspects, and there were lots of how about here, what do you do here, it was a process, it was a journey, and I learned a lot. I'm still learning.
Very, very painstaking. And I think it also helps to explain something that I know Jamshid and I get questions from people all the time, because you go on the internet and you see people saying they have stem cell therapy going back to, 15, 20 years, right? And we kind of say, if they really have it, they need to show that they have done all of this. Right. And if they don't have publications, if they don't tell you what's going on.
Yeah. What are these stem cells, right? That's very scary.
Yeah. So we have many questions coming in. I think one immediate one is people have asked, what about immune reaction to allogenic cells?
Yeah.
How do you address
that? So immunosuppression is required. So that's, and this is aspect that we needed to learn. We have some studies going on in the lab with larger animals to study the immunosuppression. For sure, as with gene therapy, right? Even if it was not a cell that's different, in particular for recessive diseases, right? You're bringing cells that express something that you don't have. So immunosuppression is required. What we don't know is for how long. So that's something, and then, it's always like that scale. What's worse? And I think that's a decision. If the cells are proven safe, and it gets established, and someone has muscular dystrophy and would like to try it, what is the risk benefit, right?
Yeah.
Of having, if long-term immunosuppression is required, what's worse, my condition or the long-term immunosuppression? Mm-hmm. So we don't know that, right? So these are things that, even though we have crossed many boxes, we just don't know. So one of the goals here is to have, in these first trial patients, the first few patients will be, the muscle will be collected during immunosuppression, because you want to know in the best case scenario, right? So if after we remove the biopsy, the engraftment's there. But then in future, once we have established that, would like to, for example, stop the immunosuppression and collect the muscle a month later, 2 months later, right? So what I can tell you from mouse studies, we have done, because human to mouse is hard to do, right? Because it's immunosuppression, but we have done studies mouse into mice and then immunosuppression, the same immunosuppression. And we know it works, right? This was the first paper was 2008. But in more recently, we removed the immunosuppression of one group and leave them for 2 months, well, at least several weeks. Are the cells gone? And the answer is no, they're still there. Well, maybe less. So we don't know exactly, these are, you can do to a certain extent in the mice, not quite the same, right? But one way that I can view, this is again, the first product, right? Because we need to start somewhere. But one way I envision is that that would be ideal instead of going autologous. Autologous, again, it adds so many layers. So one way of reducing the pain of the immunosuppression or the hurdle of the immunosuppression is that once we show these cells are safe, come with a bank of iPS cells. Then we can say, my HLA is XXY. What do you have for me? Like if you're in a drugstore, right?
Right.
So you're going to have a bank of 15. Which one is the best match for me?
Yeah.
Like you would do with blood banks, right?
Yeah.
Or donuts.
That's an interesting idea. Yeah.
So that, I think it would be more feasible than going autologous because it's still, even though you have to generate more products, you can generate 20 more, but it's not 100 more.
Customized, right?
500
Yes.
more, right? So I feel that would be the best in the long run, but that's the future.
Yeah, no, that's an interesting idea. So we have several questions relating to the fact that in the case of FSHD patients, you're transplanting into a patient who has muscle cells that are producing DUX4. So one question, which I think you partly addressed with your model, is does that endogenous DUX4, is it toxic to the engrafted cells? And then I think people are thinking along those lines, would you combine that transplantation with a DUX4 suppressing drug like losmapimod or something?
No, that's a very important point. Yeah, so that's what I tried to convey during the talk, because this was the main question we had. Okay, they engraft in the first group, right? Treat with DUX4, or induce injury with DUX4, I should say. Expose DUX4, transplant, everything is good. But what happens when you bring DUX4 back, right? And that's what we did in those 7+7, 7+14. And the myofibers didn't seem to be gone. They seem to be resistant to DUX4. I like to say that, with a grain of salt, because it's a mouse model. We never know exactly what the cells will do in the human context, right? But it's the closest we can get to a muscle environment expressing DUX4 at the moment, right? So from what we see, that re-exposure, the exposure of the engrafted cells to DUX4 actually make them regenerate even more. We didn't see, because we could think that, they could start, losing or seeing some, degeneration. So the muscle area, we could see clearly the areas that had donor-derived engraftment, they look healthy with less fibrosis, while certain areas that didn't have the donor-derived engraftment look sick. So the idea is that the cells keep healthy and you have stem cells and they're going to take over, hopefully, or at least, you can't say it's going to replace. It's very hard to promise something like this, but they have the advantage of being healthy, right?
Yeah.
So hopefully they would.
Okay.
Along those lines, you did not see a fusion of the engrafted cells with the endogenous mouse stem cells, right? to form a hybrid muscle, human-mouse muscle.
Right. We do have some fusion, but nothing obviously that we could see it. Because what we have seen is that, because it's muscle, right, they do fuse. We can't avoid that. But we also have de novo formation of myofibers. So, right. Yeah, the fusion is an aspect. So if it's fused, what's going to happen, right? Yeah. So we didn't look at that, we didn't really scrutinize that aspect.
Right. So there are a couple of questions here asking if the transplantation were to be done into muscles that are really very advanced in their degeneration, very fibrotic. Do you know what that, do they still have the ability to repair muscle that's that?
Thanks for the question, because actually I realized I didn't answer the last question about combining therapy, right? This brings me to a point where we are going very soon. I think the cell replacement is ideal because, if it works, you have functional muscle, but obviously it would benefit depending on the disease and depending on the stage of combining therapy. So the idea of blocking DUX4 and having a cell transplantation, I think is a win-win situation probably, right? Mm-hmm. The same when you're talking about what would be the state? And this is a discussion that's almost philosophical that I have every single day with my colleagues, because that was the point of which muscle you're going to target, how we're going to start, right? And I always can tell, well, I want a muscle that's damaged, because if the muscle is not damaged, the cells have no reason to stay. In the same time, if it's too damaged, what's going to happen, right? So that image that I have in the beginning, that's kind of the extreme, right? I mean, there is no muscle architecture there. So we haven't transplanted in that level, right? What I would say is that we need the level, we need damage. Whether we can revert a muscle that's really advanced, probably not. Mm-hmm. If it's too advanced, I think we would like to target muscles that are on their way. Mm-hmm. I think it's very hard, I'm not going to be promising to revert a muscle that's gone, because it'll be almost like a miracle. But what I can envision is that if a muscle is in really advanced but not gone, we could see if, treating with, a combined therapy, antifibrotics, for example, right? Yeah. Can I make that muscle healthier? Receive the cells, right? Yeah. So this is something we're studying now. We just got an NIH grant to study.
So that raises a good point, that it's important to understand also what's happening with fibrosis, and if we can reverse that at the same time that other therapies might
prepare the muscle for the cells, right? To be receptive.
Right. So it sounds like the cells would be injected directly into muscle. Is it also feasible in the future for them to be given through an infusion? I mean, can the cells cross
Yeah, so we have shown that in mouse-to-mouse studies. We have focused mostly intramuscular, because we feel you have to go step by step again, right? So that's something that we're going to focus, because that's the next, one of the next goals for us to understand, right? Because it's great if you can do intramuscular, and if it's working, there are small muscles you can target, and if you can bring local benefit, that's great. But obviously, for disease like muscular dystrophy, you want to target as many muscles as possible, right? I would say probably what we have done, we would prefer intraarterial versus intravenous, because of the fear of cells being trapped in the lung. You don't want that to happen, right? So in mice, we have done IV, and it's okay, but not. You don't want that in patients. So the intraarterial, still not as systemically as would want, right? I mean, it's systemically, but not everywhere. But you can target several muscles, one injection, right? So that's what we are gearing up for.
Okay. And then you might have touched on it way back in the beginning, but I also have lost track of. So the cells, the way you envision it, the allogenic cells would come from a healthy donor. And what type of cells do you start with? What's your preferred starting cell type to develop these iPSCs?
So we don't really have a preference. I usually also I would say probably a muscle cell is better, but all our iPS cells in the lab are skin biopsy, and this particular one is cord blood.
Cord blood.
And it makes muscle beautifully. We love this line. Everybody wants to work with this line.
Okay.
So I don't think that is a preferred tissue of origin. And there are some initial studies indicating that even though, in early passages of iPS cells, the tissue of origin, might have some impact. Once the cells have passage several times, they become all the same. So there is a paper published many years ago. We haven't really looked at this carefully. We look kind of line by line, but from our experience, it doesn't really matter. Not that I have looked too many tissues. I have looked skin, muscle, and blood.
Yeah. Okay. And then there was a question that says, if you have a spontaneous mutation, can your family be a donor? And of course, what you're saying is right now you're not looking at cells from patients, because I'm sure people who read our news have been reading about ideas about taking your own cell, that's the autologous transplant, and fixing the genetic defect in the test tube and then putting that back in. But there is a company that is working on that, Vita Therapeutics. So this whole area of iPSC-based cell therapies is getting very active. That's all I can say.
Definitely. No, I mean, we have published several studies of autologous in animal models, of course, right? Yeah. So I find, as I stated in the beginning, and I'm aware of other companies, that it's very appealing and it is attractive. But I think first you really needed to show in a safety and that we actually can generate reliable cells. I feel that now I can go back to if there is you have to think about how this will be, the economic aspect, reimbursement, all that. I know I did found a company, but this is all a new enterprise for me, right? I don't really get into those details, but these are things that people ask, how much it's going to cost, what's going to be how much can be paid by health insurance. And we have done it for a product that can be used in many patients, and it's expensive, right? So if you're going to do from one patient, if the patient has the way to fund that, but it's really expensive. That's all I'm saying, right? But I think as you establish the procedure, as like phone, right? It's like a cheap analogy, but things, once you know how to make it, right, it becomes, and the whole field is just progressing. Now we have the procedures, all the SOPs, we just have to add the gene correction. So I'm not saying we're not going to go autologous. I just find that we want to reach as many patients as possible, and we find this is the fastest way. So we really appreciate your explaining the strategy and all the considerations that go into the different approaches. So this was an incredibly enlightening talk and very exciting. So we are at the end, way past actually the end of our time, but thank you so much for sharing your research with us. I think it was incredibly exciting. Thank you. Thanks for having me.
All right. Bye.